Okay students, today our topic is food test, particularly food nutrient test. You know, different type of nutrients are present in the food. Carbohydrate. Here we can identify the main sugar component monosaccharide, and a polysaccharide. Other subfood nutrients are proteins, and third is the principal nutrient is the fat or oil. Carbohydrate monosaccharide generally identified the chemical reaction. This is called the Fehling test. Fehling test. Polysaccharides, particularly starch, are identified, boiled starch are identified by using iodine test. Proteins are identified by Xanthoproteic test. Fats or oils generally easily identified when we soak the paper by fat or oil, then the paper will be translucent, and when we see through this paper, we can easily observe the sunlight can pass through this paper. That is the translucent paper, normal rough paper, non-transparent paper transformed into translucent one. Monosaccharide. First, the food identification is monosaccharide. What is the main monosaccharides are principal monosaccharides: glucose and fructose. By Fehling test, glucose will be identified, fructose will not. Why? Why we can identify glucose but not fructose? Glucose contain the functional group aldehyde, CHO group. This CHO group easily converted into COOH form. This reaction, this process helps the glucose to identify by changing its color when we add Fehling reagent. What happens when we take some glucose in test tube and Fehling reagent is added and it is heated? What color appears? Initially, before heating, the color is blue-green. But after heating, the chemical reaction is performed and the color is changed. The color will be brick red. So what happened? Take a test tube. This is the test tube. We take the solution of sugar solution, glucose solution. In this glucose solution, we add Fehling reagent. We add Fehling reagent. What is the component of the Fehling reagent? It will be discussed in the higher class, but for our memorization, it consists of two: Fehling A and Fehling B. Fehling A contain copper sulfate and Fehling B generally contain sodium potassium tartarate. These two compounds are mixed and added. The total content will be converted into blue-green color. It converted into blue-green. But When it is heated gently, heat is applied. When it is heated very cautiously, then this blue-green color is converted into brick red color. This blue-green color converted into brick red color. Converted into brick red. This is the specific identification test of glucose. Here, the aldehyde group converted into acid. What happened in initial stage? Fehling solution, the copper ion provide the blue color. But after reaction on heating, the copper ion make a salt, copper salt of tartarate, and a copper oxide is produced. This impart the brick red color of the solution, and this aldehyde group converted into acid in nature, reducing and converted into acid. This is but fructose. What happened in case of fructose? In fructose, after heated with Fehling reagent, the color of the test tube will not be changed, because due to the presence of the keto group. Ketone is a non-reducing end, and ketone do not convert it into acid directly in this case. So fructose cannot be identified easily. This is the first test of monosaccharide. Then another more complex sugar, this is polysaccharide, is a starch. Starch is the iodine. This is the iodine test. In this test, the iodine solution plus potassium iodide. Iodine solution plus potassium iodide are mixed and in water 100 ml. This mixture is added in starch solution. When suppose this is a test tube, starch solution is added and it is heated, and then iodine plus Ki solution is poured in it. This is when after heating, this is converted into blue black. Converted into blue black. Blue black color. This is the main identifying property for the test of starch. Suppose you take a bread. Bread is the main component of starch. The main constituent of the bread is starch. Here, when potassium iodide and iodine solution is added, then the brick red color appeared in this spot where the reagent is added. What happened? In this case, after heating, this starch molecule is a more complex polysaccharide, starch. After heating, it is break into two components. One is amylose and amylopectin. Amylose and amylopectin. The amylose is a more or less coiled structure. When the iodine is entrapped here, then the color of the iodine is converted into, looks like a blue-black appearance. This is the identification test of starch. Third test is protein test. Protein. What happened? You know, protein food is the mainly composed of amino acids. and 21 amino acids are interlinked by peptide bond in various combination. Most of the protein, all of the protein contains few amount of aromatic amino acid. Aromatic amino acid like tyrosine, phenylalanine, and tryptophan. These three aromatic compound easily identified by a reaction. This is called the Xanthoproteic reaction. The reaction is called the Xanthoproteic reaction. What happened in this reaction? In this case also, you take a beaker. This is a test tube. In the test tube, some amount of egg albumin, egg white is called albuminous protein, egg albumin is taken, and concentrated nitric acid is poured drop by drop. Concentrated nitric acid is added drop by drop and heated, and from the lower side apply heat. The Bunsen burner, very cautiously. After heating, the total content will be converted into yellow color. It is converted into yellow color when heating. This is yellow color appearance due to the presence of aromatic amino acid. This yellow color may be converted into another orange form when this conversion converted into orange. In what condition? When ammonium hydroxide or other NaOH, ammonium hydroxide is added. In this reagent form a nitro compound. This nitro compound when precipitate, the yellow color converted into orange form. That is why it is called the Xanthoproteic reaction. This is the main protein identification test, and by this method we can identify the amino acid that containing phenyl group. Phenyl group, the benzene ring also present, and the phenyl group provides the color of the yellow color is converted when nitric acid is added, and it is converted also into orange color when ammonium hydroxide or NaOH is added. This is the protein test. And in the initially I told you that how fat or oil will be identified. Take a non-transparent paper, poured 1 drop of oil, the oil is soaked and the paper became translucent. When we can try the sun through this paper, translucent paper, we can easily identify to see the sunlight. Sunlight can pass through it. This is the major identification test of the lipid or fat. Then if we take a food, and obviously all food generally contain majority 90% of water. So water easily identified, water H2O easily identified by cobalt chloride. Cobalt chloride paper when soaked in a open fruit that contains water, the cobalt chloride blue color converted into pink. This color conversion From the by observing the cobalt chloride paper, we can easily identify the presence of water in a food materials. Other is the minerals. Minerals also present sodium, potassium, calcium, magnesium, boron, molybdenum, zinc. Different micronutrients and macronutrients also present in all foods. When food is burned totally in presence of oxygen, ash is observed. When we analyze the ash in different instrumental procedure, spectroscopic matter, all the constituent micronutrient can easily identify.